Hello, my name is Lucy Williams, and today I'll be telling you about a project to investigate selection in pangenomes using haplotype blocks. This is joint work with my advisor, Brendan Moomey, and with a frequent collaborator of ours, Joanne Mudge. Brendan and Joanne have done some really interesting work over the years developing theory and practical tools for pangenome analysis. But this is our first project specifically looking at natural selection using pangenomes. I'll start out by explaining the problem in the traditional reference genome setting. As sequencing technology has improved and research groups and governments have undertaken massive whole genome sequencing projects, we now have the data to be able to measure selection pressure not just for a single gene, but across an entire genome, such as in the figure currently shown on the screen. From left to right, we see every base position of human chromosome 2, which contains 242 million base pairs. The height of the bars shows what's called the selection coefficient, which gives a measure of the selective pressure at each position. With data like this, we can see areas of a genome that have experienced recent natural selection at a very granular level. In 2018, a group of researchers proposed a simple way to compute the heights of these bars. Let's say that we're interested in the selection coefficient at the 121 millionth base pair in this chromosome. At this location, and the regions around it, there's some specific DNA sequence that dominates in the population. But within certain subsets of the population, there may be slight variations, each spanning different sections of the genome. We can compute the selection coefficient for each of these variants efficiently, and then we simply set the selection coefficient of our position to the max over the selection coefficients of all variants covering it. As computer scientists, what we're interested in is figuring out how to quickly compute all of these variants, so that biologists can use as much data as they want without slowing down their analysis. In 2018, Kanha et al. Proposed to use a pattern called a maximal perfect haplotype block for these variants. In general, a haplotype block is a set of DNA variations that tend to be inherited together. And these maximal perfect haplotype blocks just give a precise definition of what a haplotype block is. To find the blocks, we need to take in a set of sequenced genomes for k individuals, with n SNP calls each. So we can represent the data as a k by n binary matrix. In this small example, we see that k is 4 and n is 6. We could build this matrix from a VCF file, which is used to store sequence variations. To find our haplotype blocks, we look for subregions of rows and columns where each row is exactly equal and can't be extended by adding more columns or rows. Let's see what this means using an example. These two rows are indeed equal, and so they satisfy the first condition. There's nothing to their left, so they're left maximal. On the right, we couldn't add another column to the block without breaking equality, so they are right maximal. And we can't add either row. So yes, this is a block. What about the blue region? It satisfies equality and both left and right maximality, but we can add a third row. So this is the block, but the original two rows were not. As you can see, these blocks can overlap, and there can be a lot of them, even in a small example like this. So a naive algorithm that just checked every single possible block, sort of like what we were just doing, would be too slow. But the good news for the reference case is that there is a fast algorithm to find these blocks. Now that we've seen what it means to measure selection using haplotype blocks in the reference case, let's move on to the pangenome case. For us, a pangenome is a representation of all of the genetic information for a species. Here we see an example of how we might represent a pangenome for a very simple organism with a very short genome, for which we've seen only two versions, one with a G in the middle and one with an A in the middle. The graph we're using to represent it is called a compressed de Bruijn graph. And the basic idea is that this graph represents sequences by their unique parts and by their overlapping parts, according to a parameter k. We call any part of the graph where the sequences diverge a bubble. And in this work, we define these bubbles to be a sort of pangenome SNP and model them as two related nodes with sides 1 and 0. In this example, we only have one SNP, which we label SNP1, and so we label our top node as 1:1 and our bottom node as 1:0. And in this case, the bubble in the pangenome only represents a variation at one nucleotide. But in general, these bubbles can represent longer runs of variation, and we can even have more complex graphs where certain parts of the graph are accessible by some genome sequences, but not by others. Here's an example constructed from about 1,000 SARS-CoV-2, CoV-1, and MERS genomes, zoomed in on the part of the graph that's responsible for making the spike protein, which is the target of the Pfizer and Moderna vaccines that we've all been hearing promising news about. Remember, this graph is just representing the pangenome SNPs, the bubbles, in the de Bruijn graph. We see an edge in this graph between two nodes if there's at least one individual in our sample population that followed one bubble edge and then later passed through another bubble edge. So what this figure is saying is that in our data set of about 1,000 individuals, there isn't really just one version of the spike protein gene that we could represent as a linear reference. There's sort of three main variants, and even within those three variants, there are subvariants. So now we have an idea of what a pangenome SNP graph is. Let's think about what it means to measure selection on the pangenome. In the reference case, we could measure selection at each base pair position. In the pangenome case, we'd want to measure selection at each SNP, which we can't represent as a bar graph before. But one way we've been visualizing it is by coloring our SNP graph nodes according to the selection coefficient that we compute, where the stronger the selection, the darker red the node will be. And according to our data, in our method, there is indeed selective pressure on some of the variants in the spike protein. In order to actually compute these selection coefficients, we adapt the method of Cunha et al. From the reference case to the pangenome case. For every SNP side node, we'll find all variants that cover the node and set the selection coefficient for that node to the max over all the variants covering it. For example, in this data set, there are two variants that cover SNP node 74, side 0, in the middle of the figure there. So just as before, the task is to compute all of the variants. And we'll define those using a maximal perfect pangenome haplotype block, which now is based on a set of paths through the graph instead of a binary matrix. And just as before, I think this is easiest to explain with examples. Let's consider the bright green path and the bright blue path. To check that these paths form a block over the subpath that we've highlighted, let's look at each condition. Yes, each of these paths follow the subpath exactly. Here, left and right maximality are trivial, since we clearly can't extend the subpath left or right, which actually for this representation of the graph is up or down. But there's a third path, this olive green path, that follows the subpath exactly as well. And so the answer would be no, that just the green and blue paths do not form a maximal perfect pangenome haplotype block, but with the third path they do. In our work, we give the first algorithm to find all pangenome haplotype blocks from a data set. We use a data structure called a suffix tree, which is built over one long string of text. So far, we've been thinking of these input paths as living on the graph, but the key insight is that we can instead think of these input paths as living in a text string, and then use efficient algorithms from the text processing world to find the blocks. For anyone who's interested, I describe the algorithm here, but what I really want to point out is that it ends up running in linear time with respect to the input, which means that it should be fast enough to use in practice. We also provide a general pipeline that can be used to investigate selection in the pangenome. First, we build the compressed de Bruijn graph, then we build the SNP graph. Next, we use our algorithm to find all pangenome haplotype blocks, and we compute the selection coefficient for each block. Finally, we assign each SNP node the maximum selection coefficient over all the blocks covering it. As you saw before, we did apply that entire pipeline to a SARS-CoV-2 dataset, and we found positive selection on some of the variants in the spike gene. It's good to see that the spike gene is under selective pressure, because that means that the virus can't just change these parts of its genome to escape the vaccine without negatively affecting its fitness. It's not shown in this picture, but we also find other regions of the virus pangenome experiencing selective pressure, so those could possibly be alternative or additional vaccine targets. We also applied our method to a yeast pangenome built using 200 strains that were also studied in a 2018 Nature paper. To create this figure, we went through that entire pipeline to compute selection coefficients, but then restricted the data to just one yeast strain that happened to have a lot of recent selection. This strain is isolated from a sake starter, and the genes where those large bars, those high selection coefficients that you see, occur in the genome, match to genes from a bioethanol producing yeast strain from Costa Rica. And those genes are likely critical to alcohol tolerance. So it makes sense that these genes would be important to the sake strain. To conclude, I'll go through the takeaways from this work and talk about some future directions we want to pursue. We provided a method and a proof of concept for computing selection in pangenomes using haplotype blocks. In the future, we'd like to look at developing algorithms for different pangenome representations that are also widely used in the field, like colored de Bruijn graphs. We're also interested in looking more deeply at the statistical power of our analysis. One concern we have is that for the pangenome, while our initial data set may contain thousands of individuals, each variant might only be contained in a few individuals. So the dataset may be too small to draw conclusions from. Finally, we're interested in looking at how to adapt our method to the case when some regions in the genome were not successfully sequenced and data may be missing. Thanks everyone for your attention. Please feel free to contact me or take a look at our implementation on GitHub. And here I'm just showing information about 2 papers that we've published or will publish on this topic. Thank you again.